Ralph Weissleder was already mentioned. He's one of the co-PIs of our CCNE project here, and he's also the director of Harvard Medical School Systems Biology program. So, Ralph?
Let me just correct that. I'm not the director of Harvard's Systems Biology. That's Mark Kirshner. I'm over at Mass General and run the Systems Biology Center there. Just bear with me for 1 second. So good morning and welcome to this year's CCR Symposium. My presentation will focus on some of our work on using nanomaterials as diagnostic tools in the era of molecular medicine. And I recognize that this is a mixed audience, so please bear with me as I provide some broader background slides and occasionally not go into as much detail as I would like for engineering communities. So as was mentioned before, nature and our human body uses a variety of natural nanoparticles, complex protein assemblies, HDL, high-density lipoproteins, low-density lipoproteins as shuttling mechanism, cellular organelles in the nanometer size range with extraordinary capability, or sophisticated biological machines such as phage make what we synthesize in the laboratory, the synthetic nanoparticles, dwarf in comparison. So irrespective of what these natural nanoparticles can do today, they also inspire us to import some of these capabilities in synthetic nanoparticles. There are some principles which I'll show you some examples of that we typically bank on. So synthetic nanoparticles have often ideal properties, in particular high surface areas, which often can be ideal for targeting and for drug delivery, as was mentioned. Often these materials can be made to exhibit pharmacokinetic behaviors which are far more superior to small molecules. The modular design allows very rapid development. They can be made as multimodality agents with combined diagnostic or therapeutic capabilities. They can be designed with complex functions as switches, docks, enhancers, and so on. But probably most importantly, after pharmacokinetics, is the ability to use multivalency to create nanomaterials with very high affinity targeting capabilities. So multivalency having several sites or using several sites of attachment for targeting properties. So let me show you one example of that. So as one does a typical surface plasmon resonance experiment, flowing small molecule over a target that it recognizes, typical on and off rates are very, very fast. As one puts these small molecules on nanomaterials and does exactly the same experiment, the kinetics here change fundamentally. The on rates are somewhat slower, and often these nanoparticles don't even come off anymore. They stick like molecular Velcro. Another way of looking at this, a specific example, is this hold ligand that has a pretty pathetic KD of 8 micromolar for FKBP, fork-head binding protein. When this material is bound onto nanoparticles, the affinity increases to 25 nanomolars. So 2 orders of magnitude. So this concept of multivalency has 2 practical implications. Number one is we can take drugs, small molecule drugs, which are not very useful currently because of lower KDs, and convert them into high affinity binders, higher KD compounds. And number two is we can take nanomaterials and derivatize them with small molecules to achieve new functions on these nanomaterials. So we have used both of these concepts for two major applications. One is bioimaging, using nanomaterials to highlight early cancers as we image the body. And then at the cellular or tissue level, use multiple colors, if you wish, to differentiate benign from malignant lesions, all the way down to the cellular level to understand the biology of how cells interact with each other. The other broad aspect where we have used these concepts of multivalency is in the design of molecular nanolabs or in vitro diagnostic devices. So for the next 20-25 minutes, I will show you 2 or 3 examples in the bioimaging field where we have taken some of these concepts to make new agents, new imaging technologies to identify, pinpoint, characterize, and diagnose cancer. And then I will follow up the last 10 minutes on some of the very recent developments in this molecular nanobiology. So in terms of, from a biology perspective, we mostly study how primary tumors develop and how primary tumors interact with a whole number of host cells, either from innate immunity or adaptive immunity. And over the years, we have created nanomaterials shown here in red that bind to or are activated by these specific target molecules. So I'll show you a couple of examples. Number one, nanoparticles targeted to primary cancers to identify cancers within the body, use of nanoparticles to read out therapeutic efficacy, and nanoparticles targeted to tissue, to tumor-associated macrophages to better understand their function, their stimulatory and inhibitory function. So let me go back to that introductory slide. So one of the first experiments that we did a couple of years ago was we asked the question whether small molecule modification of magnetic nanoparticles, which we had taken into patients at that time, based on Fe2O3, Fe3O4 magnetic cores derivatized with dextran coating. So we asked the question if these materials derivatized with small molecules would show different in vitro behaviors when screened against cells. More recently, this has gotten much more sophisticated in our ability of putting entire DOS library of these particles onto these particles. So the important point is as one screens these now entire libraries of nanomaterials against different cell types, for example, cancers against normal cells to identify tags that target cancer cells, or cells such as shown here in red, you probably can't see this, but resting macrophages from activated macrophages, we were able to identify compounds that specifically target the same cell but in different states of activation. So it's pretty amazing that small molecule modification can actually target subcells. So just some examples. So this is our nanomaterial, this magnetic nanomaterial, where we measure the uptake in cells in resting macrophages. This was our best screen for resting macrophages. This was a hit against activated macrophages, and in these resting macrophages doesn't show uptake. But importantly, as we stimulate macrophages, there is much more uptake no matter how we stimulate LPS, OX-LDL, GM-CSF, or co-cultures of tumor cells and these macrophages. So for the first time, we have now materials that actually target not just all types of macrophages, but specifically activated macrophages or resting macrophages. So some of these compounds were then taken into cancer models to ask the specific questions. When injected in vivo, does this compound here, AMTA, targeting activated macrophages, target tumor cells, tumor-associated macrophages, fibroblasts, and so on? So the experiments that we did is we started out with GFP cancer models, a number of different GFP cancer models, injected these nanomaterials, took the cells out, did extensive FACS analysis, and to make a long story short, this is our starting material. When injected intravenously, more than half of this compound actually ends up in tumor cells by itself. And remember, this is just a dextran-coated nanoparticle. Only half of it targets macrophages. So this goes against the paradigm that nanoparticles just end up in macrophages. However, the small molecule derivatized agent is over 95% specific now for activated macrophages. And in other models, such as a CT26 colon cancer, Tyler Jacks's KRAS lung cancer model, or soft tissue sarcoma model, this compound here, AMTA, is highly specific for tumor-associated macrophages. Now, why am I showing you this? I'm showing you this because for the first time, we now have the ability to inject these compounds and rapidly in different tumor models actually start to interrogate how in vivo, how these macrophages behave, what their kinetics are, how they interact with tumor cells, whether they phagocytose tumor cells, and so on. Because these are fluorescently tagged and magnetically tagged at the same time, it also gives us the ability to visualize these materials in entire animals, to look at the three-dimensional distribution of tumor-associated macrophages in this KRAS sarcoma here implanted into, or not implanted, but induced in the hind leg. And as you can see, so these tumor-associated macrophages don't just happen throughout the entire cancer, they happen in hotspots. This is the popliteal lymph node, but this is within the cancer, so there are hotspots within cancer. Let me go on to a second example, another common example of what we develop. We're very interested in developing technologies that will ultimately allow the clinicians to detect cancers before they become widespread, become very malignant, i.e. in their pre-malignant stage. So one specific example that we have targeted over the last couple of years is pancreatic adenocarcinoma. There's about 20,000, 28,000 new cases diagnosed in the US each year, and most of them are diagnosed at a very late stage. So the clinical challenge is to identify high-risk individuals and screen them before these cancers actually develop and when these cancers are still curable. So we use different approaches here. One is genomic profiling. The other one is taking patient samples and throwing nanoparticle libraries, small molecule libraries against these in the hope of identifying compounds that specifically target these earliest forming cancers, and then to convert this into imaging agents so that we can identify these lesions in vivo. So one specific example, using a mouse model of pancreatic ductal adenocarcinoma, we used phage display technology to identify phage that bind to these cancerous cells and did negative selection on normal pancreatic cells. And to make a long story short, we identified about 30 different hits that had high enough affinity and also specificity to prompt us further investigation. One of these hits here, peptide 27, by the way, I should say that a number of these target known biomarkers of pancreatic cancers, whereas others were totally unknown. So the one I'm going to show you is totally unknown. As you can see, as we take this peptide here, it beautifully separates normal pancreatic parenchyma from cancer cells. And it targets a new protein, or a protein that was previously not associated with pancreatic cancer, plectin-1. It's one of those intermediate, or one of those proteins that bind intermediate cytoskeletal proteins. And what happens in cancer cells, it gets reshuffled from intracellular compartments onto the cell surface. So these peptide sequences were then used to decorate the nanoparticle surface. And I'll just show you one specific example where in a mouse we injected, so these mice now bear pannons, intraepithelial pancreatic neoplasm, the precursors to full-blown pancreatic cancer. An intraoperative view here, the pancreas is shown here by this pink tissue. And as Dr. Grudzinski mentioned earlier today, in this type of intraoperative setting, when these particles get administered in a near-infrared channel, they can be used to highlight these 100-micron sized lesions all over the pancreas. So there are actually, in these mice, there are hundreds of these pancreatic intraepithelial neoplasm. As you can see here, the targeted nanoparticles home beautifully into these very, very, very early forms of cancer, shown here by this brown stain. So brown is our nanoparticle, dark blue is the pannon lesion. So this is one of the first examples of how one can use these nanomaterials to detect basically pre-neoplastic lesion clinical problem lesions. Another approach, the last imaging approach that I want to share with you, are smart sensors. So these are somewhat different. We build them again on nano materials. These can either be nanoparticles or polymeric scaffolds, but they work differently. So here we're not necessarily building binding agents, but rather tethering fluorochromes or other reporters through peptide chains to report out specific enzyme activities associated with cancers. Now, for imaging, this is a very important concept because it leads to amplification. Because when we inject these materials, we no longer just look at their biodistribution, but really at their local activation through specific enzymes. So over the years, we have made a number of these different probes, catepsins, MMPs, UPA, caspase, and have used them in different publications, this together with Tyler Jacks in PNAS in 2005, to identify lung cancers. But the example that I want to show you is a more recent one, where we have used an MMP12 sensor to measure specifically MMP12 activity in another cell type, in eosinophils. Up to very recently, there was no imaging agent out there to image eosinophils. We did this in this particular example, not for cancer, but for another disease, and that is asthma. And with the specific idea of ultimately imaging eosinophils and eosinophil activity or MMP2 activity in asthmatic mice. And why are we doing this? This doesn't seem to work. There we go. We're doing this because at the end of the day, what we envision is not to take tissue out and put it under the microscope, but essentially putting the microscope inside the mouse or ultimately into the patient. So what we're showing here is mouse bronchoscopy coupled with confocal microscopy. So in these live intubated mice, we're putting a 300 micron confocal microscope, as you can see here, into their lung to image the distal airways and acini in a live mouse. This is technology which is currently already in patients. So this, coupled with the molecular probes, now allows us to specifically watch certain cell types. So in a control mouse, this is a non-asthmatic mouse, we don't see anything because there are no eosinophils, versus in an asthmatic lung, There are tons and tons of these eosinophils. So a specific example where we, for the first time, see specific cell types. Conversely, at the macroscopic resolution, we're capable of reading out this information non-invasively from the outside as well. Now, all of this can now be used to start asking questions like, does specific treatments work in these mouse groups? So this is still with invasive bronchoscopy and BAL, and this is with non-invasive imaging. So one gets to molecular, basically non-invasive molecular readouts. So very, very powerful and enabling technology. So let me move on in the last 10 minutes to yet another very interesting era, explosive growth and great potential, and that is nanoscale sensing. So for this to work in the clinic, it has to be primarily fast. Clinicians don't, including myself, don't have time to wait for results. Has to be cheap and ideally multiplexed and very sensitive. So we believe that, and of course there are many different new technologies out there, and you'll hear about some of these technologies later this morning. So the one technology that we have developed in our laboratory is based on these magnetic nanoparticles that I've shown you in the beginning of my presentation, for one specific reason, and that is we believe that magnetic resonance technology is very powerful even for in vitro analysis, because it doesn't necessarily require samples to be purified prior to making these measurements. So this could take out an entire step, lengthy step in the analysis pathway. So how does this work? Again, the principle is based on magnetic nanoparticles, but unlike the magnetic nanoparticles that many of you use for cell separations, what we measure is a different effect. We actually measure the effect that magnetic nanoparticles have on surrounding water protons. And what we measure is really the decay rate by which water protons decay in vivo. Now, it turns out that this decay rate is much faster in the presence of one of these magnetic nanoparticles. So in other words, if one had a beaker full of water and measured this relaxation time, it is much, much faster if there's only one nanoparticle in there. And that's basically what we measure. Another way of looking at this is that one magnetic nanoparticle will affect billions of water molecules, and that's what we measure, i.e. there is an inherent amplification built in, billion-fold amplification. So over the years, we have developed different assay formats. The simplest assay format is magnetic tagging, where we take magnetic tags, very similar as in cell separation, put antibodies on them to target them to specific cell types, to tag specific cells magnetically, and then ask questions, how does the water behave around these to identify these cells? Another one is through magnetic relaxation switches. So here the concept is one takes two small nanoparticles, makes them into an oligomer, and by virtue of doing this, it changes the relaxivity significantly. So the relaxation time is even faster in this state than in this state. So it's a switch mechanism. And then there are other incarnations thereof, antibody-mediated magnetic-assisted amplification strategies. So despite all of this, until very recently, one of the biggest problems with this technology for more widespread use has been we still needed a magnet, essentially, or an NMR machine to read out this specific behavior. So in a hospital, it's easy. We have tons of these machines around. But not everyone does, and certainly not in Africa and developing countries. So what we decided to do and what we asked, can we develop technology, handheld-based sensors that incorporate the same NMR principles, but on a handheld scanner? And over the last couple of years, Hakko Lee in my lab, Don Heham at SEAS over at Harvard, and others, we have built one of the first handheld NMR machines. So this entire machine is 4 centimeters across, incorporates a number of different microcoils to actually sense drops of blood that we put on there, microfluidics back here, and the whole system is about 4 centimeters across currently. Importantly, the entire circuits for RF generation and for listening can be incorporated very cheaply and printed on here. Microfluidics can be used to mix these components and to separate samples. And most importantly, and I believe this is the jewel of this entire system, is we were able very recently to incorporate the entire RF circuitry, all of the electronics that drive these huge NMR machines, clinical NMR machines, on a 2 by 2 millimeter CMOS chip. So this is currently the world's smallest NMR machine. It weighs less than 100 grams. It's about 60 times more sensitive than the most sensitive NMR machine out there. So we have used this to ask questions, biological questions, specifically with respect to circulating cancer cells. Are they present in the blood of patients? Can they be used as early biomarkers? How similar, different are these compared to primary tumors? Have they undergone EMT? Do they express metastatic signature, something that we need to be really worried about in patients? Can they be used for profiling of signaling pathways or mutational analysis even? Or can they be used to measure drug efficacy? So I'll just show you a couple of tantalizing slides. So first one is detection threshold. How many circulating cancer cells can we actually detect in blood drawn from patients? So currently, the detection threshold is somewhere around 3 to 5 cells. So very low numbers. Now, cancer cells don't happen just in the vacuum, but they happen in a sea of leukocytes, they happen around red blood cells, and all these other cells that we won't detect. So what's the detection threshold in whole blood in cancer patients? It's somewhere around 5 cells currently. So there is tremendous potential in further developing this technology to get down to the single cell detection level to determine whether circulating cancer cells are present in a given patient. Importantly, in the same sample, because we have multiple channels for multiple different readouts, we now can ask questions. By the way, the detection here was done via CD326 or EpCAM. We can ask questions like, what other markers do these express and how well do they behave? So if we have developed methods for measuring EGFR on these cancer cell lines, HER2/neu, and these are just calibration curves of magnetic measurements against Western blots. I should say for the Western blots, we used 100,000 cells. For the T2 or NMR measurements, we used 10 to 100 cells. So ultimately, we envision that one has these integrated multiplex chips where we ask a number of different questions like, are there circulating cancer cells? What do they express? Are there other soluble biomarkers present? To ultimately allow us profiling such as these, are these markers present, yes or no, to come up with a malignancy signature in patients. So the one example that I've shown you is specifically this technology to detect cells. Because one can also use the magnetic relaxation switches, it can be used to read out DNA sequences, mismatches, SNPs potentially, interrogate proteins, or even measure small molecules. And because everything is based on NMR technology, these measurements can be made in blood, in biopsy samples, or even in sputum without further purification. So this becomes a real point-of-care bedside technology. So just 2 more slides where we have taken this technology now to a huge problem worldwide, and that is TB detection. So our goal ultimately is to have a handheld sensor where we take sputum that either has or doesn't have mycobacteria targeted with these magnetic nanoparticles, and then perform measurements. Now, one of the little twists that we incorporated here in this newer design of these devices is we incorporated the ability to actually wash the samples. So because ultimately we want to be very sensitive, detecting 1 to 10 to 100 bacteria per 510 cc of sputum. So what we have done is, into the microfluidics, we have put filters in, so magnetically tagged bacteria, but also free circulating nanoparticles get flown through here. The nanoparticles by themselves flow through. The bacteria get arrested here and then resuspended for measurement. So this is all integrated onto the same chip. Just some examples. So these are bacteria here. This is the filter. And so we have determined that one needs about 3 to 4 washing steps to yield or to achieve maximum T2 signal. And again, very interesting results. So we're currently able to detect about 10 bacteria, 10 TB bacteria per about 5 milliliter of samples. So this is orders of magnitude below the current WHO detection threshold. So let me just summarize here. So I believe that I've shown you that nanoparticles can be extremely versatile materials for biological measurements. One can impart unique cellular targeting capabilities by exploiting multivalency concepts. This can be used for early cancer detection. It's applicable to a number of other diseases. And we believe that new classes of nanosensors will be extremely sensitive, fast, and ultimately cost-effective altogether. This will hopefully enable molecular medicine, personalized medicine, and medicine, better medicine in developing countries. So finally, let me acknowledge the many, many people who contributed to this work, the PIs in my lab, in particular Hako Lee, who did a lot of the work on the NMR on a chip, Lee Josephson, and other people, as well as the many collaborators from MIT and Harvard. So I also like to thank the NCI for supporting our CCNE and some of these projects. Thank you very much.